Hello, everyone, and welcome to AdAlta's investor briefing, which follows the news that the first participants in the AD214 extension study had been dosed. My name's Jane Lowe. I'm Managing Director of IR Department, and it's a pleasure to support AdAlta on Investor Relations. I'll also be your moderator for this session. Joining me today is Dr. Tim Oldham, who's AdAlta's CEO and Managing Director. Good morning, Tim.
Good morning, Jane, and good morning to our participants who I can still see are still rolling in. I'll let them have a little bit more time to get in before I commence my remarks, I think, Jane.
That sounds good. And I'll just set up some housekeeping whilst we're waiting for people to roll in. So the format for today is that Tim will quickly run us through a few slides which summarise some of AdAlta's recent announcements and progress. And after that, we'll open up the floor to Q&A. If you'd like to ask a question, you can do so using the Q&A ribbon in the bottom of your screen. With that, though, I will now pass the floor over to Tim. And Tim, I'll ask you to start your screen share whenever you're ready and get us on the road. Thank you.
Thank you, Jane, and good morning, everybody. Thanks for joining us today. We've made some enormous progress on our lead program, AD214, over the past 2 months. That's been summarised in around 4 major ASX releases over that period of time. My purpose today is really to bring that all together, to summarise the enormous progress we've made to de-risk AD214 as our lead asset, to improve its partnerability, and fundamentally to increase our confidence that we have a genuine solution to help patients with debilitating and fatal fibrotic diseases. Okay, so many of you will have heard me talk at length about our quite extensive pipeline of projects today that cover not only fibrosis, but also cancer and immunotherapies, all driven by our iBody technology platform. Today, though, is all about AD214, our lead asset, treating degenerative, progressive and fatal fibrotic diseases such as idiopathic pulmonary fibrosis. And to kick us off today, I'd like to introduce Bill Van Nierop. Bill is one of the 500,000 reasons why we're developing AD214 for fibrotic disease. Bill is actually one of the lucky ones. He is an IPF survivor, or an idiopathic pulmonary fibrosis survivor. He was first diagnosed with IPF back in 2015. As the scarring took over his lungs, reducing their flexibility and reducing his respiratory capacity, Bill took on the challenge of raising awareness for IPF. It is a rare disease and it's not well known, but it is absolutely debilitating for sufferers. So despite the impairment of his respiratory function, Bill first took on a 700-kilometre walk through central west New South Wales to raise funds for the Lung Foundation of Australia and for IPF awareness. He then took on a 2,200-kilometre paddle of the Murray River in 2018, all of which were enormously successful in raising awareness for this debilitating disease. As his disease progressed, Bill was lucky enough or unlucky enough to need a lung transplant, lucky enough to actually receive one. That happened during COVID. And now, despite the fact that he is now a survivor, his passion for raising awareness for IPF is undimmed. On the 28th of August, he is commencing his second long-distance kayak, this time down the Murrumbidgee River. And I commend you to support Bill's efforts to raise awareness for this debilitating disease. The website where you can donate is at the bottom of this slide, and we're thrilled that Bill is continuing to work to build out the awareness for this debilitating disease. This is why we get up in the morning at AdAlta to develop AD214. There are approximately 1,250 people like Bill diagnosed in Australia every year. Most of them are not as lucky as Bill. There are 500,000 people diagnosed or living with IPF around the world. And on average, they survive for less than 4 years from diagnosis. They're mostly older people, 55 or older. They're treated with 2 current therapies that generate sales of $4.3 billion a year, but they have a limited effectiveness and serious side effects. And now we're just talking about lung fibrosis. There is fibrosis affecting almost every other organ in your body. So for the Bills of the world and those that are not as lucky as Bill, we are trying to develop a new therapy for lung fibrosis and other fibrotic diseases using a novel mode of action that provides new hope for these sufferers of this debilitating and fatal set of diseases. So a reminder of where we are today. Over the past 5 years or so, we've really built a strong value proposition around AD214. It's a first-in-class molecule targeting a validated what we call mode of action or disease pathway. And being first-in-class creates competitive advantages and attractiveness to big pharmaceutical companies who will ultimately commercialize this molecule. We've demonstrated preclinical efficacy, i.e. reductions in fibrosis, reductions in collagen deposition. Collagen is the molecule that creates the stiffness in scar tissue, and various other markers of disease progression in multiple animal models of fibrotic diseases, most notably in lung and kidney fibrosis. We can manufacture this molecule at scale. We've successfully completed an initial phase 1 study showing that the drug was well tolerated in healthy volunteers. We've recently released data, which I'll talk about in a moment, showing that not only is the efficacy we've seen in animal models appears to translate to models of disease at clinically relevant and clinically feasible dosing regimens. And we have a really strong intellectual property position through 2036 with orphan drug designation in the US, which confers significant regulatory advantages for our eventual partners. We've just commenced an extension study of the phase 1 program. We dosed the first participants last week in that study. That's a really important program that I'll talk about also in some more detail. We're accelerating existing partnering discussions and adding new partners to that conversations. We had an amazingly successful meeting at Bio Industry Conference in Boston in June. And we're continuing to prepare the molecule so that potential partners or other financiers can take over a ready-to-roll phase 2 clinical program. So let's turn to the new data that we've released over the past 6 to 8 weeks. So the first and potentially most important piece was the demonstration that AD214 is potentially efficacious at a clinically viable or convenient dosing regimen. Now, I'll step through this gradually so that we build up the picture. As shown in panel A of this slide, and as I mentioned previously, we had shown that AD214 was efficacious in mouse models of fibrotic diseases. That was the important tick to enable us to go into phase 1 clinical trials. Panel B summarizes that in those phase 1 clinical trials, we demonstrated a safety profile, and we understood the profile and duration of AD214 to block its target receptor in humans. And we knew, therefore, the length of time when AD214 totally or partially blocked the CXCR4 receptor, which is its target. What we didn't know was the piece of data that links those two important insights. We didn't know what level of receptor occupancy had been achieved in the mice because it was really difficult to measure. And we didn't know what level of receptor occupancy was sufficient to modulate disease, therefore. As a result, while we knew that we needed to dose AD214 intravenously no more frequently than every 2 weeks for clinical convenience, we didn't know whether that was going to be sufficient to impact disease progress. So what we did was take a model of a fibrotic process that we could study in the lab. We looked at one of the mechanisms by which AD214 works, and this is the inhibition of the migration of inflammatory cells into a wound area, such as a lung or a kidney. And we tested different concentrations of AD214 on that model to see A, how it inhibited the migration of those cells, and B, what level of receptor occupancy was achieved. And what we found was that we didn't need 100% receptor occupancy in order to maximally inhibit that fibrotic process, that model fibrotic process. What that told us, therefore, was that the levels of receptor occupancy we'd seen in the phase 1 clinical trial at 1, 2, and potentially 3 weeks after dosing were likely to be sufficient to be effective in modulating disease. In other words, the clinically convenient dosing regimen that we'd been testing in phase 1 clinical trials now had the evidence to show that it was likely to be effective in treating disease. And that was an important link that gave us great confidence in moving forward with both the intravenous administration of AD214 at these 2 weekly dosing intervals, and therefore progressing to phase 2. And it answered a big question that our partners had been asking. So from there, we've now commenced a phase 1 extension clinical study, where the objective here is to increase the maximum dose that we have our safety profile for to 10 milligrams per kilogram, which lines up with the maximum likely effective dose that we've identified as a result of the studies I just discussed. This phase 1 extension study is important, not only because it establishes that safety profile, but it results in a avoidance of the need to do that dose escalation in phase 2, which reduces the cost of a phase 2 study to our partner. It will better inform the dosing levels and schedules for phase 2, because we will now have much more accurate pharmacokinetics and pharmacodynamics profiles for the drug. And it enhances our partnering process, because this additional data helps to address some of the known and potential questions that our partners have been or are anticipated to ask us. It also maintains momentum for product development in a way that generates new data rather than some of the other investments we could have made in this drug, which would have been all about spending money without any, there were important spends like manufacturing material for phase 2, but they don't add any value by de-risking the program. So this is the most important, most valuable investment we could make to further de-risk AD214, and we're delighted that that program is now back in the clinic with our first participants treated successfully last week. So that brings me on to the partnering landscape, the third piece of the pillar that I wanted to cover today. Now, AD214 is in that zone between phase 1 and phase 2 ready. And as you can see from this slide, there's been a very dynamic and active partnering landscape for molecules at this stage of development, ranging from AbbVie's purchase of DJS antibodies in October last year at preclinical stage for $255 million through to Genentech's licensing of a Kinexa asset for $80 million up front and $620 million in contingent milestones in August last year. And so you can see that just the upfront payments for these assets are multiples of our current market capitalization. There's a very active partnering landscape with multiple companies looking for multiple assets in their pipeline. While our primary objective is to help IPF patients and other fibrosis patients like Bill Van Nierop, that won't happen unless we can convince pharmaceutical companies, and pharmaceutical companies are out there willing to pay money to acquire and license these assets and then take them through to commercial development. And what this table shows is the confidence that we have that this is a dynamic and attractive partnering landscape. Now, how does that translate to AdAlta's partnering initiatives? Coming out of the bioindustry partnering conference in Boston in June, this is what our partnering pipeline looked like. We have 21 companies in various stages of discussions with us around AD214, ranging from they've started a commercial evaluation on the assumption that a phase 1 asset moving into phase 2 is within their strategic landscape, through to active technical evaluation, which means that we're talking not just to the business development team, but also and search and evaluation team, but also to the scientific teams under a non-confidential environment, and through onto confidentiality agreements where we're starting to really deep dive into the data that is not in the public domain. So we have a really robust partnering pipeline across the world. This includes both global companies as well as regional champions, particularly in Japan and China. And this gives us confidence that not only is there global interest in IPF assets, there is active interest in our IPF asset, AD214. And then bridging back to our broader strategy, out-licensing AD214 is really important to us to ensure that we can finance the phase 2 program. But it is only one of 3 partnering initiatives that we have underway at the moment. We are also continuing to out-license our iBody discovery platform, aiming to do more collaboration such as we have with GE Healthcare or with Carina Biotech that partially or completely finance discovery programs. And we're also evaluating complementary near to the clinic assets to bolster our clinical stage pipeline, which is, as you will all be aware, is the key to unlocking value in biotech companies, particularly in the current environment. We've expanded our advisory network to help us prosecute these transactions, and I'm really pleased with the development of the partnering pipeline as it stands today. So a recap of our corporate strategy at the moment, 4 pillars to that: realise the value of AD214 through partnering, and that has been significantly enhanced as a result of the demonstration that AD214 could be efficacious at clinically practical and reasonable dosing regimens, and that we're now back in the clinic. Our second strategy is extending our ICAR programs through additional collaborations similar to the one we have with Carina Biotech. Our third pillar is progressing our GE collaboration in PET imaging for immuno-oncology. And our fourth theme is continuing to invest in our iBody platform to ensure that it remains globally competitive. And this includes the potential to add additional clinical stage assets that would help bolster and expand our pipeline. So in summary, as a company, as an investment proposition, we continue to be underpinned by our iBody platform across all of our programs. We're advancing significantly AD214 into a $4.3 billion market for just its first indication. But in addition, don't forget, we have 4 other programs with 2 other collaborations running already. We've demonstrated that we can develop products through the clinic. We've demonstrated our partnering expertise. We have a underappreciated market capitalization, in my view. Most CEOs, of course, will say that. But we have some significant blue sky inorganic growth opportunities through our 3 partnering programs at the moment. We do have additional news coming over the next 3 to 6 months, particularly through the readouts from the AD214 phase 1 extension study. And so that will continue to confirm, we hope, the de-risking and the increasing confidence that we have in the AD214 asset. And with that, Jane, I will hand back to you to moderate the Q&A session.
Thanks, Tim. So with that, I'd now like to open the floor to questions. Thanks to all those who've already dropped some into the Q&A. Reminder, if you'd like to ask a question, do so using the Q&A ribbon at the bottom of your screen. Okay, so we do have a few here on partners in particular. So what are you hearing partners like about AD214?
Partners are attracted to AD214 for a number of reasons. The first is that it is a novel mode of action. We've seen a number of reasonably significant clinical trials fail at phase 3 in recent times. And so pharma are getting discouraged about those particular modes of action and like the fact that we have a different approach to treating fibrosis. That's also beneficial because we can be used in combination with many of the other modes of action as well, and fibrosis is likely to be treated as a combination drug. So for a pharma company, that de-risks, that reduces commercialization risk because there'll always be a place for it. The animal models of fibrosis have always been challenging. They're highly variable and they don't translate well. We've had that experience ourselves. what pharma really like is the work we've done to study the mode of action in what we call ex vivo or in vitro models, so in a lab, in a test tube. And this is actually proving to be more powerful than some of the animal models we've done. The animal models are necessary but not sufficient. Pharma is really appreciating the work we've been putting into the in vitro studies. And most compellingly was the recent data looking at the link between receptor occupancy and efficacy that helps us get confidence in the dosing schedules. That was a big tick for us and not something that everybody has. We've got adequate patent protection, which is always one of the very early questions that they will ask us. And so they're probably the three big things that are really attractive to this molecule. And of course, we have a great safety profile as well, in contrast to many of the small molecule drugs, which are accompanied by nausea, vomiting, diarrhea. One of the marketed drugs makes you photosensitive to sunlight, for example. So many patients can't tolerate those for more than a year. So our safety profile is really attractive as well.
Good. I recall Bill Van Neurup talking about those same side effects with his experience of using those drugs in the past. So actually, I did have another question here on, and you briefly touched on it here, but the recent receptor occupancy data. So you did recently release some data on receptor occupancy for AD214. So what has the response been to that?
So far we've been able to share that with a number of pharma companies at Bio, where we already had a confidentiality agreement in place, and it really cemented that work we've been doing on the mode of action studies. Several of them had been asking us for some time, can you link your efficacy with the phase 1 data? What receptor occupancy do you need to be effective? What receptor occupancy did you see in the mice? When we went back to them with this data, one of them said, I really appreciate that you've actually listened to what we've been saying and you've generated this data. That's really powerful and really helpful for us in our evaluation of the molecule. So that was just one example of the kind of response that we're getting. And so it's proved to be an experiment that was very difficult for our team to do. I'm very, very proud of the team for the effort they've put in to make this actually work. They're very difficult experiments, but it's definitely been worth it. We've actually also filed some of the results were quite unexpected, and so we've been able to file some new patent applications around some of the findings as well.
That's great. Okay, so noting the current cash balance and cash burn, are we trying to organise any upfront cash payments from potential partners?
So good question. Ultimately, the structure of any transaction will be, a balance of value versus how do we actually progress the asset. You can see from the slide that I showed earlier that the vast majority of IPF licensing transactions come with significant upfront payments. So we would anticipate that we would be eligible for some upfront payment of some kind. We would also anticipate and expect that our partners would take on the burden of the phase 2 costs as well. So certainly our objective is plan A for funding our phase 2 program is through the partnership, either through the upfront payments or through the costs that will be taken on by our partners. Plan B is there are actually a number of project financing vehicles out there where investors will take on the risk of a specific clinical trial. So plan B would be to progress down that route, and we are maintaining that momentum as we progress our partnering discussions. But yes, absolutely, the objective is that it's time for AD214 to earn a return on investment. The investments we continue to make in AD214 have to meet some very rigorous criteria. They have to generate new data that will help our partnering initiative. They have to be relatively low cost. And the clinical trial is by far the most significant investment we plan to continue to make in AD214. And then the other work we're doing is to ensure that the necessary enabling activities to support phase 2 are in place. So this means we've got manufacturing slots for the drug manufacturing booked. We have toxicology slots for the extended dose toxicology studies booked. We're not spending the money on those, but we've made sure that they're booked and in the calendar so that a partner doesn't have to start from scratch, because the lead times for these can be quite long.
Got it. Okay, thank you. So this was always designed to be a quick briefing today, quick update for investors. I've got one other question in the queue. If you'd like to get one in, now's your chance. Use that Q&A function in the ribbon at the bottom of the screen. But for now, we'll run with this one, which may be our last. So an important one, what data do you expect to have from the AD214 extension study by the end of this year?
So the study's got a really interesting design where the patients will receive 4 doses of AD214. The first 3 are at 2-weekly intervals as per the original phase 1 study. The results of that first wave of doses we expect to have before the end of the year. And that will reconfirm our safety profile, confirm and extend the receptor occupancy profile we have for the drug following multiple doses. All the participants will then receive a 4th dose a month after their 3rd dose. And that 4th dose may end up falling around about Christmas time, early January at the latest, which means that the top line results of all of the study will be available in the first quarter of next year. At this stage, February at the latest. So we will definitely have the results that match the original phase 1 study by the end of the year. And then we'll have the add-on results that we've been able to use design refinements to achieve additional insights from this trial. We'll have that by February next year.
Excellent. Well, as a shareholder myself, it's really exciting to see you back in the clinic. I'm sure it's exciting for you as well. Congratulations on getting there and dosing the first participants. Some terrific news.
Yeah, thanks, Jane. It's been a really exciting and a really busy last 2 or 3 months. We've got lots of work still to do, but we've made massive progress, increasing our confidence in this asset in the last couple of months, and certainly reducing risk for partners. And we're seeing that reflected in the quality of conversations we're now having. So look forward to further updates very soon.
Excellent. Well, I think that might make those closing comments. So thank you, Tim. Thank you, everyone, for joining. And we look forward to keeping you across AdAlta's progress. I'll invite you all now to disconnect.